Hi everyone, I am Asenad with the topic of hybridization techniques and chemical synthesis of oligonucleotide. Let's get started. Hybridization techniques. Hybridization techniques refers to various methods used in molecular biology and genetics to combine or merge genetic materials from different sources. These techniques are essential for studying gene function, manipulating DNA and producing genetically modified organisms. In biotechnology, hybridization techniques play a crucial role in various applications, including genetic engineering, gene expression analysis, and diagnostic assays. Here are some hybridization techniques commonly used in biotechnology. First one is the recombinant DNA technology. This technique involves combining DNA molecules from different sources to create combined DNA molecules. It's typically involved the use of restriction enzymes to cut DNA at specific sites, followed by ligation to join the desired DNA fragments. Recombinant DNA technology is fundamental to genetic engineering, allowing the insertion of foreign genes into host organisms to produce desired products or modify their characteristics. Next one is the DNA microarrays. DNA microarrays, also known as gene chips, are solid surfaces, typically glass slides or silicon chips, with thousands to million of immobilized DNA probes. These probes are single-stranded DNA molecules that are designed to be completely to specific target sequence. By hybridizing labeled DNA or RNA samples to the microarray, researchers can determine gene expression levels, identify mutations, or analyze DNA variations. Reverse transcription polymerase chain reaction or RT-PCR is a technique used to amplify and detect specific RNA sequence. It involves the conversion of RNA into complementary DNA or cDNA using the enzyme reverse transcriptase, followed by PCR amplification of the cDNA. RT-PCR allows the quantification of gene expression levels and is commonly used in gene expression studies and diagnosis. Next one is the ligase chain reaction or LCR. LCR is a DNA amplification technique that uses DNA ligase to join two short DNA probes that hybridize to adjacent target sequences. By repeatedly cycling the reaction, the target DNA sequence is amplified. LCR is useful for detecting specific DNA sequencing and is particularly advantageous for its high specificity. Next one is the southern blotting. Southern blotting is a technique used to detect specific DNA sequences in a sample. It involves digesting DNA with restriction enzymes, separating the resulting fragment by gel electrophoresis, transferring them to a membrane, and then hybridizing them with a labeled DNA probe to detect the target sequence. Next one is the northern blotting. Similar to the southern blotting, northern blotting is used to detect specific RNA molecules in a sample. The RNA is separated by gel electrophoresis, transferred to a membrane, and hybridized with a labeled DNA or RNA probe complementary to the target RNA sequence. Next one is the fluorescent in situ hybridization or FISH. FISH is a variation of in situ hybridization that uses fluorescently labeled DNA or RNA probes to detect and localize specific sequences in cells or tissues. It is widely used in cytogenetics to analyze chromosomal abnormalities and gene expression patterns. Next, we are moving to the chemical synthesis of oligonucleotide part. An oligonucleotide is a short chain of nucleotide, the building blocks of DNA and RNA, typically made up of around 2 to 50 nucleotide units. These molecules play important roles in molecular biology. Genetics and biotechnology. Oligonucleotides are often used in laboratory experiments for tasks such as DNA sequencing, polymerase chain reaction or PCR, and gene synthesis. They can also be used as therapeutic agents, such as in the development of antisense oligonucleotide drugs for treating genetic disorders. The chemical synthesis of oligonucleotide involves the step by step assembly of nucleotide units to create a custom DNA or RNA sequence. Here are the basic steps involved in the chemical synthesis of oligonucleotide. They include detailation, coupling, oxidation, and capping. Detailation: after each nucleotide is added to the growing chain, the DMT group on the 5' OH hydroxyl group of the newly added nucleotide must be removed before the next nucleotide can be added. The removal is achieved through detailation. This is the diagram showing the detailation in detail. Detailation is a crucial step in the chemical synthesis of oligonucleotide. It involves the removal of the protecting group known as a trityl group, often referring as TR, from the 5' end of the oligonucleotide strand. The step is necessary to expose and activate active 5' hydroxyl group for further nucleotide coupling reaction. Here is how detailing typically occurs. This is the